Hi guys, let's start with proteins. They are also part of the biological molecules chapter. So let's get started. Proteins are compounds of carbon, hydrogen, oxygen, and nitrogen. Okay, and they can also contain phosphorus and sulfur as well. So a jumble of these elements make up the proteins, and we'll see how that goes by. And they are all polypeptide chains of amino acids. Okay, now what are amino acids? Amino acids are organic compounds, okay, containing carbon, and they're used as monomers, okay, like many small units connected together to form a large polymer, okay, in the making of all the different types of proteins. Okay, pretty simple, pretty clear. Okay, and let's move on to amino acids, the basic structure of amino acids. Okay. Now this is the basic structure of amino acids. Now as you can see, we have four main parts of this structure. Okay, let's highlight that. This is the first part, the carbon and hydrogen, they are fixed like this. The second part, okay, is the carboxyl group, okay, or carboxylic acid group, okay, and this is the amine group, this third part. And the fourth part, okay, it's a free radical, and it can be changed. Okay, there are different types of free radicals that can attach here, and hence they make different types of amino acids. Okay, now on the basis of free radicals, okay, there are 300 plus different amino acids. Now we divide them into two categories. Okay, the first category is proteinous, the second is non-proteinous. Okay, now proteinous amino acids, okay, there are 20 of them. Okay, they're used to make all the proteins in all the life forms, okay, be it animals or fish or plants or any living thing. Okay, they used to make all the proteins. Okay, the rest, okay, are 280 plus proteins. Okay, they are non-proteinous proteins, and they are the most are naturally occurring. Okay, for example, thyroxine and dopamine. However, some are synthetic. Okay, they're man-made. Pretty simple. Now a main concern will be with proteinous amino acids, because obviously we are learning this under the chapter of biological molecules. Okay, we need to consider the life forms. Now let's see a few examples of proteinous amino acids. Now the simplest amino acid, okay, and it's the most common as well, it's glycine. Okay, need to remember the name, and this is the structure. Okay, now here you can see the free radical is hydrogen. Okay, hydrogen is attached, and everything else is the same. We have the amine group, we have the carboxylic acid group, okay, and we have the CH. Okay, now the second most common and simplest, okay, the second most common and simple is alanine, and it has a CH3 radical attached to it. Okay, and everything else is the same. Okay, now let's go on to a few general types. Okay, now this box here represents this the basic structure, this thing. Okay, this, because this is the same. Okay, we have replaced this with a box, and connected to the box are the R groups, or the free radical groups. Okay, so these are the free radical. Okay, so we have an example of an acidic R group. Why am I saying acidic? Because it has COOH, as you can see. This here is an alkaline R group, NH2 group. Okay, and we have a polar R group. Okay, why is it polar? Because we have a dipole forming. Okay, because oxygen is electronegative, it's highly electronegative, and hydrogen not. Okay, so we have the dipole in this direction. And this is the polar R group, amino acid with the polar R group, and this is an amino acid with a non-polar R group, which is actually alanine. Okay, as we studied here. This is the structure of alanine. Okay. Now we need to study a very important property of amino acids. Okay, and that is that amino acids are amphoteric in nature. Amphoteric means having dual nature. Okay, that means they act as both acids and bases. We'll see how that goes. Now, in alkaline solution, okay, if you look at alkaline solution, if I add an amino acid, okay, any amino acid, okay, irrespective of the R group, okay, if I add that in an alkaline solution, okay, what will happen? Okay, it will reaction irreversibly, and it will dissociate. Okay, the COOH here, okay, it donates a proton, okay, or you can say it dissociates to form H plus ions, okay, and that's a property of an acid. Okay, and resulting we have the remaining COO negative ion, okay, and that's attached to the C. Okay, so it's acting as an acid in alkaline solution. Okay, let's see what acts as an acidic solution. In acidic solution, now what ends up happening is that it gains a proton. Okay, it accepts a proton and forms NH3 plus ion. Okay, and since it is accepting a proton, accepting an H plus ion, okay, that means it is acting as a base. Now in neutral solution, okay, it acts as both acids and bases. Okay, it gets a positive charge at one end and a negative charge at the other. Okay, like this. Okay, if we water, okay, if we add this into water, okay, this will happen. The COOH, okay, it dissociates to form COO negative and H plus ion, and that H plus ion we can say that it goes back to here. Okay, it goes to here to form this thing. Okay, from here to here it goes and forms the NH3 plus ion. Okay, very simple. We give a special name to this ion, this whole complex, okay, this whole thing, and this is called as Switter ion. Okay, right pronunciation. Okay, now this whole thing, okay, this shows, okay, that amino acids can neutralize both acids and bases. Okay, yes, because if I add an acid to an amino acid, okay, it will act as a base, and if I add a base, it will act as an acid. Okay, now this is important. They do not completely neutralize. Okay, it depends. Okay, it depends to some extent. Okay, so that means they can act as buffers, okay, in the blood plasma. Okay, they are helping to maintain the pH of blood. Okay, the buffers are that substances that resist a change in the pH. Okay, and they kind of regulate, okay, the pH of the blood. Okay, pretty clear, pretty simple. Okay, now let's see how actually do we make a polypeptide chain, okay, from the amino acids. Now what ends up happening, okay, what do we do? Okay, let's see. Now to make a polypeptide chain, okay, let's first understand how do we make a dipeptide, or two amino acids connected. Okay, now how are they connected? So if I have these two amino acid molecules here, okay, you can see this first one and this is the second one. Now in close proximity, okay, in certain alignment, okay, what ends up happening is that we take the OH from the COOH, okay, group, and we take the H from this NH2, okay, and that this water molecule is removed. It's a condensation reaction. Now what ends up happening is that this carbon here forms a bond with the nitrogen, and it kind of like appears like this. Okay, here we have the bond here, CN and O and H. Okay, now there are two important bonds here. Okay, the peptide bond specifically is only the carbon nitrogen bond. Okay, carbon nitrogen bond is the peptide bond. But however, if we talk about the amide linkage, or the amide linkage, that is this whole thing, CONH. Okay, this is the amide linkage. And similarly we can form tripeptides. Okay, then we can form polypeptides. Okay, similarly we attach many amino acids, okay, together. They form a long chain. Okay, they act as monomers. Okay, and through multiple peptide bonds they are connected together to form a very long polypeptide chain, and hence they form proteins. We'll study about that next video. That's it for this video. Thank you for watching.